Okay, so as stated, the title of my talk is Reverse Genomic Prediction, Identifying Important Traits in Breeding Populations. And as Andreas said, I'm the professor of plant breeding methodology here. So I'm going to sort of focus this talk on a lot of work related to how can we improve plant populations using modern tools. And so, of course, here's the timeline of a crop. Something in the past has been modified to be the crop that we all know today in the present, and we're moving towards the crops of the future. As a plant breeding professor, this is really where my focus lies chiefly, or at least the applied part of my job focuses on how can we move from the present into the future. So first, some history. The classical approach for a breeder to move from the present into the future, or to move plants forward, was to use traditional quantitative genetics. So here's a figure of, it's actually a fairly recent figure from a work we did last year, showing distributions of plant height in some maize fields. And it's not too important to pay attention to this. I'm just using this figure as an example to show that when you have a quantitative trait, such as the height of a plant, you end up having nice distributions of the phenotype, and you can do things like draw cutoffs and make predictions about future populations based on quantitative characteristics of the current population. So a lot of traits, not just plant height, follow this pattern. Some examples that I just pulled out of my head are yield of grain, the flowering time of a plant, whether or not a plant falls over, that's called lodging, and so on. You can measure all of these in a quantitative way, and they tend to vary continuously, often because they're controlled by many genes. So the classical approach to do quantitative genetics is, like I said, to play with phenotypic distributions of a trait and use tools such as what I've drawn here, the breeder's equation, where you can actually predict the response to selection based on the heritability of a trait, which is a genetic property of a trait you can measure, and the selection differential, which is the difference between parents and children. So this was really useful and still is really useful, and it's basically how we've arrived at modern plants, at least until the last 20 or 30 years. Tools, phenotypically based tools like the breeder's equation, were almost completely what breeders use to do plant breeding and plant improvement. But then in the 90s, maybe the 80s, marker-assisted selection came along, and this involves using a marker to differentiate individuals that are better or worse at some phenotype. So here, if we have a marker, the A marker, where capital AA individuals have a lower phenotype than lowercase AA individuals, we could use that marker to do marker-assisted selection and determine, based on the genotype, not on the phenotype, which individuals are more likely to be high-performing, and we could breed with those individuals. Now, of course, you don't have to use just one marker. You could use 2 or 3 and do this marker-assisted selection approach using an index. So here I've drawn this as Y, or your predicted phenotype, can be the sum of 3 different genotypes and 3 different weights for those 3 different genotypes. So we could do this for the A locus, the B locus, and the C locus, for example. In every case, we know that a particular marker is associated with our phenotype of interest. Okay, so then more recently, since the dawn of the new millennium, breeders have started doing something called genomic prediction. In this case, we've taken advantage of the fact that we can do marker-assisted selection with 1 marker, 2 markers, 3 markers or so, and we've realized we can just do prediction using every single marker in the genome, whether or not it's associated with a trait. So the big insight behind genomic prediction is, let's use every marker to predict a phenotype from genotypes. We build a statistical model that basically is the sum of marker effects multiplied by genotypes across every marker in the genome. And then based on that sum, we have a predicted breeding value, and we can use that for making selections and doing breeding. So I can't overstate how revolutionary genomic prediction has been for breeding. But it really was adopted by animal breeders well before it was adopted for plant breeders. And I think a couple of folks in the audience took a course with me, so you guys know what's coming. If I ask why is it that genomic prediction has been more adopted by animal breeders, it's really a story of generation time. So here's a cow, an animal, or this is not a cow, it's a bull. And the bull says, it's going to take you a long time to phenotype me. That's me instead of moo. So this is a Holstein bull, and he's being bred for milk production. And you obviously can't measure the production of milk coming from this bull. So to figure out how breeding works with a bull, let's tell a little love story. This bull falls in love with this cow. They're going to have a bunch of kids, and then the bull's milk can be measured, right? Because we have a mating, and we have several cow children. I'm only drawing the female children here, and we can measure milk production from each of these female children. So we're one generation removed, and we can get a phenotype on that bull. But not really, because cows don't make milk until they have children. So actually, we don't get the bull's phenotype measured until he has grandchildren. And really, in the grand scheme of things, you don't get a good measurement until a second lactation or so of his children. So it can take, on average, more than 5 years to go from the bull being born to the bull being phenotyped. And that's a really long time when you're trying to select which bulls should be allowed to have children, and they need to have grandchildren before you can even make a decision. So if we look back at the breeder's equation, response to selection equals heritability multiplied by the selection differential, this can be translated into response per year, depends on L, the generation interval. So these are equivalent formulations. And as you can see, the longer your generation interval, the less response to selection per year you have. So that really is what has driven the major changes in breeding that have resulted from genomic selection in animals. Going back to this bull, if we can build a statistical model of his phenotype right here before he's ever had any children, we can say, is he worth selecting or not 5 years sooner? So if we come back to plants, genomic selection, or the time component, is not quite as limiting in plants as animals. If I want to phenotype a maize plant, I can just grow it in the field, and about 3 months later, I will have a measurement of whatever trait I want to measure. So this, for maybe a decade or so, was a barrier to the adoption of genomic prediction in plants relative to animals. But now that's not really such a barrier anymore. Us plant folks have realized there's a lot of really beneficial uses for genomic prediction. So you can save money by phenotyping fewer plants and just using genotypes instead. You can increase accuracy by having a phenotypic measurement, which is measured with error, and a genetic prediction of the phenotype. So then you get 2 levels of measurement of a phenotype. And also there's time savings too. You can make predictions before you've actually measured individuals who might get selected. It's just not a 5-year difference. And then lastly, and a lot of what my talk today is about, is the fact that we can actually run genomic prediction in reverse to learn about the past. And this, I think, is quite exciting, and hopefully by the end of the talk, I'll have everybody on board that this is a good idea. But before I tell you how we can use genomic prediction backwards, I want to tell you why learning about the past can be useful in a breeding context. Why is it that breeders should even think about yesterday instead of only think about tomorrow? And to do that, here we are. To do that, I want to start off by telling a little bit about the story of maize domestication. So humans domesticated maize from this wild plant called teosinte about 8,000 or about 10,000 years ago, give or take a few thousand. And so teosinte is a grassy wild weed that grows in central Mexico. The seeds have these hard seed coats and they're not so great to eat, but they are edible. Teosinte is edible. And it's extant. You can find it growing today, especially as a weed near maize fields. And anyway, after humans did what they did to teosinte, it turned into maize in Deutsch, this plant that we often eat, especially in America, where I'm from. We love to eat it on the 4th of July off of a barbecue. Anyway, we can ask the question, what evolutionary processes drove the transformation of teosinte into maize? And one of the first candidates for an evolutionary process that you would think is very important is a classical selective sweep. So I'm going to draw some cartoons of different types of evolution, and this is the first one of these cartoons. The way a classical selective sweep works is, if we imagine here I've drawn a number of haplotypes or a number of chromosomes floating around in a population before selection. So you could think of these as haploid individuals, or you could just think of them as chromosomes that we don't care what individual they're in, they're just in a population. And blue is invariant DNA, that's just the chromosome, and black dots are neutral polymorphisms. So these are A's, T's, G's, and C's that have no impact on the phenotype. And then suppose there's a new mutation, this little red dot that happens to be beneficial. Natural selection over the course of several generations should fix this beneficial mutation. And when that happens, what you get is the fixation of alleles surrounding the beneficial mutation, because recombination has only broken down some of the relationship between this mutation and the haplotype where it first arose. And therefore, you have a reduction of genetic diversity surrounding the selected site. So our classical pattern of selection looks like this. If we look at diversity, you go from just background neutral level of diversity with normal noise to a giant dip in diversity in a selected population surrounding the selected site. So we can look at the pattern of genetic diversity across the genome to determine whether or not that classical selective sweep scenario that I just drew was the driving force of evolution. And the way we do that is we use substitutions, and we look at levels of diversity surrounding synonymous and non-synonymous substitutions. So first, what's a substitution? What's a fixed difference between one species and another species, or one population and another? In this figure from the last page, the last slide, the red dot is a substitution because before selection, nobody had the red dot, and after selection, every single individual carries this red dot or red allele. So if you look across a whole bunch of individuals, you can identify many substitutions in one species compared to another. And then we could categorize these as synonymous substitutions that are at a synonymous position of the genome that have no impact on the protein that's made, or as a non-synonymous substitution of any type. We could have a stop codon substitution, a missense substitution, various things that somehow change the protein. So I'm only looking in genes here for this analysis, not intergenic DNA. And then we can say what should happen if this classical sweep pattern has been driving evolution. So at non-synonymous substitutions, we should see classical selective sweep pattern, and at synonymous substitutions or surrounding synonymous substitutions, we should see no giant loss of diversity, because synonymous substitutions don't change the DNA, they shouldn't be selected. So we implemented this analysis using 23 maize individuals that were sequenced to high coverage across the Americas they were collected from. And we also did it in teosinte, but I'm actually not going to talk about that here. And then we evaluated how important were hard selective sweeps or that classical selective sweep that I drew 2 slides ago. Before I give you the answer, let's look at a few implementations of this in other species. In Drosophila, this is the first species I know of where they did this type of analysis, and they saw a major reduction of diversity surrounding non-synonymous substitutions relative to synonymous substitutions. So this said, yes, hard selective sweeps or classical strong selection on beneficial mutations was a driving force in Drosophila evolution. For humans, the pattern was quite different. Around various classes of substitution, there were similar reductions of diversity, and the takeaway from this was there is not evidence that strong selection on beneficial mutations or classical selective sweeps is a driving force in humans. And our pattern in maize is right here. It's very similar to what was seen in humans. We have similar reductions of diversity for both synonymous and non-synonymous substitutions. So this is averaging across every substitution in the whole genome that we identified. And this tells us that strong selection on large effect beneficial mutations has not been the primary driver of evolution in maize. So this was interesting. We published it a while ago. In that same study, we looked at how important has purifying selection been as a driving force of maize evolution. And so let me draw another cartoon here of a form of selection, in this case, purifying selection. And in this cartoon, we have the same general layout as the other cartoon with a bunch of haplotypes from a population, but I've added dots representing genes, or not dots, blue bars. So here I've imagined that there's 3 genes drawn in this population. And now suppose new mutations, instead of being beneficial, tend to be deleterious. Then selection will purge these deleterious mutations from the population, and after many generations of selection, we'll have no red dots left in the population, but we'll also have less genetic diversity near genes than we have outside of genes because of linkage. And sure enough, when we did that same analysis, what we see is that for both maize and teosinte, there has been overwhelming evidence of very strong purifying selection, getting rid of genetic diversity within genes. So most mutations tend to be bad, a few mutations tend to be good, but the good ones don't seem to be that good. So we had to keep looking for another type of mutation or another type of selection that might be driving maize evolution and its differences from teosinte. And for this story, I want to talk about polygenic selection, or selection on traits that are controlled by many genes, each of small effect. So here we have another cartoon. In this cartoon, imagine that these red dots are beneficial, but they all have very, very small effects. So after many generations of selection, we might increase the frequency of a few of these beneficial alleles, but probably nothing's reaching fixation unless evolution runs on forever and ever, because everything has a small effect. And we're proposing here polygenic. There's many of these things across the genome. So to study the impact of polygenic selection on maize evolution, I want to talk about a case study of a breeding population that was selected for maize silage quality. And this is actually data from a population I was tangentially involved in during my PhD at Wisconsin. My former PhD advisor, this is her breeding population over there in Wisconsin, but I didn't start working with these data until more recently. So this is the Wisconsin quality silage population, and it's a population that's been selected for 5 cycles for the quality of the silage. So this is ground up maize that's fed to cows so that you get better milk out of the cows. And they aren't selecting just on yield, they're really selecting on, they are selecting on yield, but other parameters in the selection index include how digestible is the maize when it's in a cow's stomach. And so over these 5 cycles, which each cycle is several years, and it's a total of about 20 years of selection, they've documented quite a bit of genetic gain in the population. So let's just take a look at some of the traits. This is a subset of traits that they've measured in the population. The first is the starch content, which increased over the course of the breeding program. This was expected. The next one is neutral detergent fiber in green. This has decreased. So NDF it's abbreviated as, and it'll be abbreviated that way in a few more slides also. And you can see neutral detergent fiber decreased consistently over the course of the experiment. This high neutral detergent fiber is associated with less digestible maize, so this was expected. And then the last trait is dry matter, which was not a part of the selection program. So it's expected that there was no trend for dry matter to increase or decrease during the selection regime. So back in 2015, we did a study looking at could we find genome-wide association study hits for any of these traits that were part of the Wisconsin silage mapping or Wisconsin quality silage population. And here is a very disappointing GWAS with absolutely nothing that showed up as significant when we looked for it. This is for starch content. We got this pattern repeatedly over and over again. We had no significance for any of the traits that we studied in this population. So then we thought, well, it's a selected population. We could use selection mapping where we compare allele frequencies before and after selection to find out which genes are playing an important role in the population's improvement. But no, we couldn't. Once again, we failed to find any significant associations between genes, or not genes, but markers and selection. And so this was a disappointment at the time, and we published it as a negative result and said, yeah, it could be that the population, or that these traits in the population are very polygenic. They're controlled by so many genes that we just don't have the power to identify selection on any of them. So that was where we left it at that point. More recently, but still beginning several years ago, I started working with Henner Simeoner and now my postdoc Medhat Mahmood on developing a method to address this problem of how can we understand selection in this type of population or with these types of traits where we just don't have power to identify any genes associated with that selection event. And the method we've been working on, we call it Ghat because it is equivalent to measuring the genetic gain in a polygenic way. So we measure the estimated allelic effect of every single locus in the population, and then we measure the allele frequency change over time at that locus. So we make a statistic we call delta G or Ghat, which is equal to the sum over every position in the genome of these two things multiplied by each other. And after we do that sum, we can actually easily permute the I values for either allele frequency change or effect size to have a test of significance that is statistically valid for the most part. So to come up with these allelic effect estimates, the alphas, we use a standard genomic prediction model to associate every single marker in the genome with a prediction of phenotype, and then we look at that across time. So let me explain for a moment why we consider this a form of reverse genomic prediction. So recall from earlier on in this talk that I described in genomic prediction, we try to predict a phenotype or predict a breeding value really by doing a summation over all markers of an effect of a marker multiplied by the genotype at that marker. With Ghat, we do almost the same thing, but instead of using a genotype, we use an allele frequency change between two generations. So we have today's generation or today's genotypes compared to the population in the past. And based on Fisher, this is equivalent to the genetic gain of a specific trait. So instead of using today's genotypes to learn about today's phenotypes, we use yesterday's genotypes to learn about what traits were significantly selected in the past. And as I hope you saw from the first few parts of my talk, knowing about the past can tell us a lot about how we should do our selections in the future. So let's look at how this GHAT statistic works and if it does a good job. Here I have a simulation of pure genetic drift on the left. I think, gosh, I don't even remember the parameters of this simulation, but I think we had a trait with a 50% heritability and, and there it is, and a trait that's controlled by 1,000 QTL. And I think there were 20 generations of either drift or 20 generations of selection. So when we simulate pure drift, there's of course no relationship between the allele frequency change and the effect size of a particular locus. However, when we're simulating selection, so we're selecting for high values of the trait, what we see is that larger effect loci, more beneficial loci, tend to increase in frequency, and more deleterious loci tend to decrease in frequency for polygenic traits controlled by 1,000 QTL. So that's just what we'd expect. And then when we apply our GHAT test to that, we get these results. The permutation test I mentioned is the red bar, and the GHAT value, the delta G that we actually get out of the test, is the blue bar, or the, yeah. So, so when we apply this, when we do a simulation of pure drift, you can see that our GHAT value falls right smack dab within the expected value for a non-selected trait. When we do a simulation of selection, we get a GHAT value that comes way beyond what we would expect if there were no association between frequency change and effect size. So back to this Wisconsin quality silage population I was talking about. As you can see, for these three traits I mentioned early on, starch, neutral detergent fiber, and dry matter, we applied the GHAT statistic or the GHAT test, and we found basically exactly what we expected based on the known phenotypic trend of the population. So looking at starch content, we see this relationship between allele frequency change and effect size across all genotype markers, and we get a significant p-value saying that starch was under selection in the population. We knew that because we did the selections. So this is still kind of a proof of concept that GHAT is working. We haven't learned anything new, but it's nice to see. With neutral detergent fiber, you can see that we find selection against neutral detergent fiber. So we haven't been able to find a perfect relationship between the size of our GHAT statistic and the strength of selection. However, we do have a true relationship between when we find positive selection or selection for increasing values, we get a positive GHAT, and selection for decreasing values gives a negative GHAT. And then for dry matter, which I said early on, we don't think was under selection at all, GHAT finds that there's no significant evidence of selection on dry matter. So that was expected. So this was a nice confirmation that our GHAT test seems to work. And if we didn't know that these three traits had been selected in the population, it would have provided that as new information. So one of the best things about GHAT, and this is a simulation result that I'm sharing here, is that in contrast to traditional selection mapping or genome-wide association studies, with GHAT, you get more power when you have a trait controlled by more QTL. So in this figure, moving from the left to the right, we are doing sets of simulations where the only thing that's changing is the number of QTL that control a given trait. So here we have 10 QTL, 50 QTL, 100 QTL, 1,000, and 10,000. And the power of the GHAT, or the detection rate to find true selection, just increases as you get a more and more complex or more quantitative trait. And drawn on here for comparison is traditional selection mapping. When we're trying to map specific genes, we lose power when we are getting a more and more quantitative trait. So we think these can be very complementary methods, that if you can't find specific genes because your trait is too quantitative and you don't have power to find the genes, you can use GHAT to find out, has that trait been selected at all by breeders in the past? But I also should emphasize here, this isn't a completely equal comparison. The two traits or the two tests are doing something different. Selection mapping is trying to find a selected gene, and GHAT is trying to find whether or not a trait was under selection based on all of the genes. So we're not ever going to get out from GHAT, this was a gene that was selected. Okay. So now I have another really fun biological story to tell. And this is work done by my former postdoc, who's now a research scientist at Bayer in St. Louis, Sarah Hassan. And she used GHAT to look for traits in Brassica oleracea. So Brassica is a really fun species because it's basically all of the good plants that we eat, especially here in Germany. So the coal crops are brassicas, and these include Brussels sprouts and cauliflower and what's this one? Wurtzing, I think. Is this Wurtzing? Anyway, since I've moved to Germany, I've started eating so much more Brassica oleracea than I ever used to. Kale, this is Chinese broccoli. It's a good plant. So in this case, the story of Brassica oleracea is that it was many years ago, many years ago, but not as long ago as a plant like maize, it was domesticated from a wild plant, and selection on different parts of the brassica plant led to all of these different crops that I just mentioned. So selection on terminal leaf buds led to cabbage. Selection on lateral leaf buds led to Brussels sprouts. Selection on leaves developed kales. Selection on stem, I believe this is kohlrabi. And then floral buds selection ended up in broccoli and cauliflower. So pretty neat that one species has developed all of these different plants that we get at the grocery store. And Sarah teamed up with a PhD student at the University of Missouri named Mackenzie Mabry to look at leaf phenotyping and then try to find out, have different patterns of selection led, or can we identify the different patterns of selection that led to these different phenotypes in brassica? So here's a bunch of leaf trait or leaves, and what they did is they took, I think, the fourth leaf from every plant in a big diversity panel of Brassica oleracea, and they scanned it, and then they measured characteristics such as total leaf area on all of these brassicas. So here's a bunch of brassicas broken down by their scientific name that I don't understand, but these are all of those brassicas. And here's their leaf size based on area from a scan. And I think in total, they had something like 400 different phenotypes they measured on these leaves coming out of the scans. But here we're just looking at area. So then they applied, or Sarah applied the GHAT test to find out, has there been selection on leaf area? And her results are pretty neat. So the way to read this plot is she's looking here at pairs of species and then saying, is there selection for or against different leaf sizes between these species? So for instance, here when she looks at Brussels sprouts compared to kale, she's saying were Brussels sprouts and kale divergently selected for leaf area based on GHAT. And a colored dot means there was a significant GHAT statistic, and the size of the dot has to do with the strength of, or the evidence or the level of significance in the test. So what was neat here is that she sees evidence of selection for smaller leaves in Brussels sprouts and in this guy Brassica insularis compared to everything else, and she sees evidence of positive selection in Chinese kale and in Brassica incana. For reasons that I can't fully describe, this confirmed a lot of suspicions about differences in leaf size in these two species, or not species, in these two morphotypes. I want to give next a more concrete example for me of the exciting thing we can do with Ghat, which is looking at selection on glucosinolate levels in cabbage and or in Brassica oleracea. So glucosinolates are the bitter compounds in Brassica, and they have evolved. There's a lot of research that suggests that high glucosinolates exist in Brassica oleracea as a defense mechanism against wild predators like caterpillars. And humans in general have selected Brassica species or crops that have lower levels of glucosinolates. So when Sarah applied the Ghat test across these brassicas, what she found is that there was selection for higher glucosinolate levels in most of the wild species in her study. So these three, Villosa, Rupistris, and Macrocarpa, are all wild, and relative to the domesticated varieties or the crop varieties, she sees selection for increased glucosinolates. So another way to read that is that there's been selection for lower glucosinolate levels within the crop brassicas. So that was a pretty cool result. And down here, this was a collaboration to measure the glucosinolate levels with Dan Klebenstein and Ella Katz. I think Klebenstein is at Davis, UC Davis. I don't remember exactly where Ella is. Okay, so this was exciting in Brassica, and now I want to go through one more case study applying the same sort of research to wheat. And here's a paper that was published, I believe, last year by a number of colleagues, many of whom are in Germany, but none of them are us. So I was not on this paper, but I, along with my team, with Medhat Mahmood, who I think he's in the audience, we scavenged, or what do you call it? We took the data because these great authors made it freely available after they published their paper. They said, you guys can take our data and do whatever you want with it. It's on the internet. So we downloaded their data, and thanks to these authors for providing it, and we applied the Ghat test to learn about, is there genetic evidence of breeding or of selection on particular traits in European wheat. So first, let's look at grain yield. This is a plot from this, and this research that I'm about to share was mostly done by Medhat Mahmood, who's a postdoc in my group. Selection on, or the phenotypic trend for grain yield in this population of several hundred wheat varieties grown in 50 years across Europe was for grain yield to increase steadily over time. And this is consistent with what we all know and expect of breeding. And Medhat applied Ghat, and sure enough, found very significant evidence of selection on grain yield in wheat. So this is consistent with what we would expect and not a huge surprise. But what was exciting was to take just the modern portion of that data. So here I just erased everything before, I think 2010, maybe, well, anyway, just the last few years of data, we reapplied the Ghat test without knowing anything about phenotypes from the past. And we said, can we still identify that there's been selection on grain yield when we don't have phenotypes from 1965, 75, 85, and so on? And sure enough, we still have very strong evidence of selection even when we're using only modern data. So this starts to demonstrate one of the really neat applications of GHAT is we can take a modern crop and compare it using old genotypes, maybe even museum samples that we can get genotypically, even if we can't grow those old varieties and phenotype them in their actual environment and determine whether or not there's been selection on a given trait. This wheat study I mentioned had several traits measured, I think 15 or 18 traits in total. And this is a kind of complicated plot showing a bunch of things. So bear with me as I walk us through it. Going down the middle here is the trait, and over here is just a continuation of the plot. So we have grain yield, biomass, blah, 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 and then radiation interception efficiency, green canopy, blah, blah, blah. Bunch of traits. And then we have a plot of the phenotypic trend across time. So tighter oval means a stronger correlation, and the text drawn on here is the p-value. So this bit on the left, grain yield, phenotypic trend, is exactly a plot of the correlation here over time. Then on the right is our GHAT test for selection in 3 different environments. You can ignore the environments, they're just different fertilizer treatments, but basically we always have the same trend across environments. Our GHAT test for selection using all years of phenotypes is drawn here, and modern phenotypes only is drawn here. So as you can see, we have significant evidence for selection on grain yield when we use modern and old phenotypes, but even when we use only modern phenotypes, we have significant evidence of selection on grain yield. And that tends to hold across most traits. We definitely have less power when we use only modern phenotypes, but we're still getting very high p-values or very low p-values for most of these traits. So kernels per spike. The phenotypic trend tells us it was probably selected. The all phenotypes information agrees with that. And then when we use only modern data and no phenotypic trend, we still recapitulate that yes, there's very strong evidence of selection for kernels per spike. So this was exciting, and only in the case where there was weak selection or where there was weak evidence of selection in the first place, such as on 1,000 kernel weight and resistance to stripe rust, do we start to lose significance for GHAT when we try to apply that test to only modern data and not old data. So this was fun. This was exciting, and hopefully we'll get this reanalysis of the Voss-Fels paper published pretty soon, along with this announcement of an R package that we have released to implement the GHAT method. So MedHot released about a year ago an R package for GHAT. And the paper announcing that is still on the way, but the package is ready to go. One more thing I want to share, a fun demonstration of something else we can do with GHAT is we can break down our data into different timelines. So here again is that same exact wheat data. And if we look across groups of years, we can look at changing selection trends through time. And this was a really nice analysis that MedHot came up with. So we have a plot for 4 different traits here. These are grain yield, harvest index, radiation use efficiency, and radiation intercept efficiency. And as we'd expect, for all periods of the last 50 years, grain yield was more or less positively selected. That's what we can tell by the dot with the error bars. When the error bars don't overlap with the 0, that means there's significant evidence of selection. So maybe from 86 to 95, breeders weren't paying enough attention to grain yield. But other traits really stand out as only being important during certain time periods. So radiation-related traits from 1986 to 95 were huge for wheat breeders, even though generally they weren't paying too much attention to them. And I don't know enough of the history of wheat breeding to know why that might be. And these types of things can sometimes be driven by, one particular breeder who released a lot of lines who had a particular selection protocol that paid attention to radiation use efficiency. But it's really neat to get that data out genetically from what was previously just a bunch of released lines. So this has been a fun thing to see. And we have this broken down for all 15 of the traits, but I decided I couldn't fit them on this plot. So you only have 7 here. Anyway. With that, I want to give a final diagram as I move towards wrapping up, and I did it. 45 minutes, I was told, and I'm going to be right on time, which was my goal. So wrapping up, the question is, how can we really use reverse genomic prediction? I've spent a lot of today talking about it. I think you saw I started by saying, look, we can do genomic prediction, but the past is important too. And so the way that we can use this GHAT reverse genomic prediction method for future improvement of crops is to first determine, based on past evolution, what traits are important. And then using that information, we can actively apply it using whatever modern tool we feel like, genomic prediction, GWAS, whatever, to breed the future crops that matter the most. And let me just verbally share an example of that. In maize, for many years, breeders were not paying any attention to the angle of a leaf. So maize leaves can go straight out to the side, or they can go almost vertical. And only in the last few decades have breeders looked at their maize plants and realized, whoa, all of our maize plants have almost vertical leaves. And a big part of that was that they were breeding for increased tolerance to high plant density without ever realizing that a sharply angled leaf lets a plant tolerate a higher plant density. So with a tool like GHAT, we could hopefully identify earlier on leaf angle is an important trait. We've been selecting for it inadvertently for the past who knows how many years. Let's start actively selecting on this trait to try to continue to improve the species. So I believe with that, a bunch of acknowledgements, my team here in Göttingen, several collaborators who were involved in various parts of this talk. And most of all, I want to thank all of you for your attention. And and I'd be happy to take any questions. Thank you.